Hello, everybody. Welcome to the Closer Look Journal Club. We're a group of people who are really interested in discussing topics in biomechanics. I have with me my co-hosts, Matt Bursey, Johannes Weikenmeyer, Adrian Buganza-Topoli, and Manuel Roche. And we're really excited today to introduce Maria Holland. Maria Holland is a Clare Booth Luce Assistant Professor of Aerospace and Mechanical Engineering at Notre Dame University, where she leads the Computational Mechanics of Morphology at Notre Dame Lab, or the Command Lab. Dr. Holland completed her bachelor's degree in mechanical engineering at the University of Tulsa in 2011, and then received her PhD in mechanical engineering from Stanford in 2017. And as you'll see in this talk, she's done a lot of really nice work on leveraging the toolset of computational mechanics to better understand living systems. Dr. Holland, thank you so much for joining us today. Please go ahead.
Thank you for having me. This is, I think, a really nice idea, a really nice format to have people. I guess I took it as an opportunity to sort of talk about some of the story behind this paper that we published. I think the date says 2021, but really was published in 2020. So the title of the paper is Numerical Investigation of Biomechanically Coupled Growth in Cortical Folding, and it was published in MBBM last year. So I like to start out pretty much all of my presentations with this slide, so I apologize if you've seen it before, but I studied the development of the brain, and since often my audience is not neuroscientists or neuroanatomists or anything, I like to just start with sort of a fun quiz question. So I can open the chat here. If you would like to enter into the chat your guess, which of these is a human brain? So these are all, they're actually stained cross sections of brains, all taken from this front angle and not to scale. So don't use that as a proxy. So any guesses? So a negative guess, not F or D. F, C, C. Lots of C's. Okay, C strong, E, C, E. Okay. The consensus seems to be C and E. So let me reveal the answers. A is actually a dolphin. So we're interested in the cortical folding, the development of this folded or wrinkled pattern of the brain. So A is a dolphin, and you can see that dolphins have a lot of folds. And again, these aren't to scale, so the folds aren't really smaller. They're about the same size as the other ones. But dolphins and whales actually have very highly folded brains, actually more so than humans. B is a dog. So you see these like very characteristic, strong patterns of folds, but many fewer than the dolphin. C is actually a chimpanzee. So a lot of you guessed C, and that's not too far off. These are some very close relatives of us. D is a rabbit. So I see that nobody guessed D, I think. Rabbits have basically unfolded brains. You can see maybe one or two little dimples in there. E is a human. So a lot of you guessed E, and that's great. But then as Johannes sort of poisoned the well, it is sort of a trick question. F is also human. This is a fetal brain around 21 weeks of gestation. And so I like to show this quiz to sort of engage the audience, hopefully, but also because I think it really nicely illustrates the process that we're interested in studying. So cortical folding or gyrification happens largely in the third trimester around actually a little after this image is taken, largely during gestation, but our brains continue to develop a little bit after birth as well. So this process by which the brain goes from this small, smooth object to this large, highly convoluted one is what we're interested in studying. And as I mentioned, these images are not to scale, but above I'm showing actually scaled images and 3D images. So you can again see this change in both size, but also shape that's happening. And so this is what we study. And the approaches we use to do this, we use a couple different approaches. So we do simulations of cortical folding, numerical simulations using finite element analysis. We also do analysis of brain morphology, so looking at actual geometries that we see in the brain and what patterns do we see. So again, here I have some images of developing fetal brains, and then I also have some comparative images of different, this is a human and a couple non-human primate species, which we can also investigate. And then I also do a little bit of work on just general instabilities in soft layered materials. So the brain is one example of that, but there are a lot of examples both within biomechanics and in other engineering applications. So we do some of this sort of fundamental work. And actually the things that I'm going to talk about today touch on sort of all three of these approaches, at least briefly. So this is just to give you an idea of some of the questions that we're interested in regarding cortical folding and things that we're working on, especially in our simulations. This is a figure from a review paper that was actually, it says submitted, but it was actually accepted to the Journal of Biomechanics this morning. So yay, I didn't update that citation. Okay. Like I said, we do these simulations of brain development, and there's a couple factors or facets sort of that we're interested in pushing forward, that just we as a field, not necessarily just my research group, but they're sort of the frontier of this work, and really the interesting next steps. So most simulations of cortical folding don't include any influence from the skull, the cerebrospinal fluid, or the meninges, and that could be really interesting. They could play a role in a number of different aspects of cortical folding. Most simulations of cortical folding use a homogeneous cortex, so it has homogeneous properties, homogeneous growth. But in reality, that's of course not the case. So here we're showing some of the cortical laminae. So there's sort of layers within the cortical layer that presumably have different properties and different functions. It would also be really great to include subcortical anisotropy, so from the effects of axon fibers and axon tracts. That's generally not done right now. And stiffness contrast, so the two tissues, generally speaking, we speak of two tissues in the brain, the gray matter and white matter. They have different properties. These properties are still, there's a lot of uncertainty in them, but it seems like they're actually very similar, at least in stiffness, and a lot of our simulations are not quite there yet. And then finally, brain geometry. So you'll see in our simulations, we do a lot of very simplified idealized geometries, like two-dimensional rectangles or three-dimensional blocks or ellipsoids and things like that. But really the overall gross geometry of the brain is really varied. There's areas of different, very high curvature and low curvature and even concave regions that you can see here. So these are sort of, like I said, the frontier of cortical folding simulations that we identified. And I've done a little bit of work in most of these areas, but today this talk is going to touch mostly on this question of cortical heterogeneity and a little bit on the stiffness contrast. So in particular, sort of within the domain of cortical folding and brain development, we're interested right now very much in cortical thickness. So this is the thickness of this outer cortical layer that I've outlined in white here. And cortical thickness is a biomarker of neurological health, and abnormal thickness is associated with a number of mental and neurological disorders, including autism spectrum disorder, epilepsy, schizophrenia. And so understanding why this happens, like what sort of things are contributing to the development of cortical thickness is really important. And the thickness varies across the brain, and it's sort of, so here on the left, I'm showing 12 neurotypical, fairly neurotypical. Sorry, so these are actually images of brain scans of myself and a number of my PhD lab mates. So neurotypical is maybe questionable, but so a healthy, relatively healthy adult humans. And I've colored different regions of the brain by their average cortical thickness. And so there's a color bar there. And so you can see that there's quite a bit of variety in different regions of the brain, from around 2 millimeters to around 3.5 millimeters. Actually, the range is a little bit beyond that. And it sort of seems like this is happening randomly. I don't really see a pattern when I look at it this way, but we can actually divide these regions into gyral regions and sulcal regions. So gyri being the sort of peaks, the outer, like the mountain peaks of the folds, and the sulci being the valleys. So if we divide the regions into sulci and gyri, we actually see that the sulci tend to be thin and gyri tend to be thick. So sulcal regions are on the blue side of the scale, and the gyral regions tend to be on the red side of the scale. So there's this characteristic pattern that sulci are thin and gyri are thick. This has actually been known for a long time. There's a well-cited paper from 1929 by a German anatomist noting that the structure of the human cerebral cortex is considerably different at the bottom of a fold, so that's in the sulcus, and at the peaks of the turns adjacent to this bottom. And these structural differences between the peaks and valleys of the folds are found again and again to occur in the same way at every fold. I just want to make a note, this paper was originally published in German, as a lot of the research literature was done so at that time. And so I have this image here of this undergrad, Andrew Fullwider, who is a double major in engineering and German here at Notre Dame. And he helped me translate this paper. And so we're actually going to be making that public soon for any of the non-Germans in the field who would like to read one of these fairly foundational papers in the field. So, so this is a really cool paper to me because he was sort of noting similar things that we're interested in and asking questions that we're interested in answering, but 90 something years ago. So we see this characteristic pattern in the brain of thick gyri and thin sulci, and a number of different hypotheses could be formed as to why this happens. And so one of them would be a very biological hypothesis that the brain is sort of programmed to grow this way, right? It's just maybe cells are more directed towards one region of the brain, and that becomes a gyrus, and so therefore the gyri are thicker. And this is a very biological hypothesis based on cell behavior, gene expression, and things like that. And so I had read this in the literature, and it wasn't quite convincing to me, at least not as the whole answer. And so I started investigating in the simulations that I had run before, and I noticed that in our simulations, even that had no source of biological heterogeneity, we saw this natural pattern of thick gyri. I mean, of course, they're not gyri in a simulation, but thick, outer folds and thin inner folds. And so this led to a paper that was published in Physical Review Letters in 2018 with my PhD advisor, Ellen Kuhl, and two collaborators, Alon Goyeli and Sylvie Budai. And so these I'm showing here are simulations that show this evolution over time, over folding of thick gyri and thin sulci. And we also did experiments in stretched polymers that showed, again, in this simulation where we originally had constant cortical thickness, homogeneous growth, we naturally got this evolution of thick gyri and thin sulci. And so that suggested to us that there's actually, the mechanics of folding are leading to this pattern, and it's not all just biology. Bach, back 90 years ago, had also sort of hinted at this. So he pointed out that, remember, the cortex is curved at the top and bottom of these folds in the opposite direction. And so this curvature must, geometrically speaking, profoundly influence the cortical structure. And he sort of posited that we will see that all architectural differences between adjacent tops and bottoms of folds are only products of the local curvature. Our work is supported, maybe not the all part of this, but definitely supported that this curvature and the forces that are generating that curvature are contributing to this pattern of thick tops and thin bottoms. So now I'm sort of finally getting to this paper that I'm here to discuss. So one of the first things that we did in this paper was we show that these thickness differences are a unique property or a unique feature that emerges in what we call soft systems. So these are, I mentioned, the brain has, generally speaking, two tissues. We have the gray matter and the white matter. And that's what our simulations have as well, two different materials. And if both of those materials are of very similar stiffnesses, that's what I would call a soft system. So we can track the thickness ratio over time as in different simulations. So if we have, a soft system or not. And so if we have a stiff film and a soft substrate, so that would not be a soft system, then the thickness ratio doesn't really change over time. It stays very close to 1 even as growth occurs. But if we have a soft film, so we have a soft film and a soft substrate, then we see this drastic increase in the thickness ratio before it plateaus at some point. And we can look at a certain time and compare the thickness ratio that we see in different stiffness ratios. And that's what we see here as well. So for basically a homogeneous system, if the film and the substrate are the same stiffness, we see a very high thickness ratio. And as the film gets stiffer, that thickness ratio drops. And around, by the time you get to a stiffness ratio of 10, the thickness ratio is completely negligible. And so this is a unique property that we see in these soft systems, which I think sort of explains why it hadn't been commented on before, because many engineering systems are out in this high stiffness range, whereas biological tissues, which are just in general still less studied, are more in this soft regime where we see this emergence of this thickness ratio. So what we found from our PRL paper is that our mechanical hypothesis of thickness differences yields a really nice qualitative agreement. Like we see this emergence of thick gyri and thin sulci in our experiments and our simulations and in the actual human brain. But there's sort of a caveat to this. Quantitatively, the numbers that we reported in this paper didn't agree. So these are the ranges of thickness ratios that we reported in our experiments, which range from like 1.2 to 1.6. So, gyri being, 60% thicker than their sulcal counterparts. And in the human brain, we just didn't see numbers that high. So we reported, in the occipital lobe that the difference is around 10% up to maybe 25%. And so quantitatively, these don't match. And so at the end of my PhD, I was sort of sitting with this and it rankled me a little bit, right? Bothered me. Why don't they, what else are we missing that is not capturing this quantitative agreement? So my natural hypothesis, I think it was a good idea at the time, was I thought that sulci must grow preferentially to reduce this gyral thickness ratio. So sort of mechanics wants to drive these two regions apart in thickness, and biology would rather have, the brain, maybe the cortex would function better if there was more of a constant thickness. And so there's sort of this opposing forces trying to normalize these two thicknesses. So that was my hypothesis, and I was like, great, let's prove how right I am. And so I thought of this sort of biomechanical hypothesis where there's, yes, the effects of mechanics in cortical folding on these thickness differences, but also, cells are doing their thing, gene expression, we have evidence that gene expression differs in the location of future gyri and future sulci. So there's got to be some aspect of biology in here as well. So how can we model this? And so that was the question that I sort of sat with for really a couple of years, was how can we do this? And so the idea that I had was it would be great if we could incorporate both the morphogenetic growth in the cortex, which is generally how this is modeled. So there's not a mechanical component to that growth. And if we just add in stress-driven growth in the cortex, which we've done, lots of people have done in the subcortex, if we put those two in there, we can have sort of this, the stress-driven growth would represent maybe that, maybe neurons, as they're migrating out to the cortex or something, they don't want to go into some place that's very compressed. If you think about yourself maybe moving through a crowd, you might go naturally towards a place where there's more space between people, right? I think that's natural versus trying to drive through the most crowded part of a room. So I thought we could just sort of combine these two and have this nice biomechanical model of growth. The problem with this is if we have morphogenetic growth in a confined space, we're going to generate compressive stresses. And then if you add in the stress-driven growth, we're going to get stress-driven shrinking, actually. Generally, a tissue under compression is going to try to shrink. And so these sort of coupled together, we're just going to go in a loop and not get anywhere. So the tissue is going to grow because of the morphogens, and then it's going to shrink because of the stress it's experiencing. So that's just not really going to work. Another problem with this is if we think about the cortex as being in bending, which it's not entirely, but there's some similarities here. A beam in bending has local regions of tension and local regions of compression on the inside and outside. So there's not sort of like a stress marker that's sort of consistent in a whole gyrus or in a whole sulcus. Instead, we'd have like the inside of gyri and the outside of sulci that have similar stress patterns growing similarly, and then the opposite. So I thought about this and I just like couldn't quite figure out how to do it. And then once I had some students and a postdoc, I think really had the tools to do this. So the idea that we ended up coming up with was to do curvature dependent cortical growth. So this is a map of the human brain showing the mean curvature. And so you can see this is an artificially inflated brain where you can see the whole surface. And you can see that there's a lot of regions of blue and red. But when we're only looking at the gyri, basically the visible parts of the folded brain, we only see the blue. So what I'm showing here is that mean curvature gives us a pretty good division between gyri and sulci. So gyri have negative mean curvature and sulci have positive mean curvature. And this, a lot of our analysis of the morphology of human brains is done by my graduate student, Nagihan Demirci. And so then my postdoc, Shuolun Wang, really took this idea and ran with it and came up with this curvature dependent model of cortical growth. So he defined the cortical growth as a function of mean curvature. And so I want to just be clear here, we're not sort of insinuating that cells are like sensing the curvature of the cortex around them and modifying their behavior in response to that. Rather, we're saying we're using mean curvature as sort of a macroscopic indicator of whether a point is in a gyrus or a sulcus. And so we can modify the growth based on that is going to sort of reflect whatever might be going on that would determine differential growth in gyri and sulci. So with this equation that I'm showing here, we can vary this parameter R curvature, and it sort of adjusts the dial on what kind of growth we have. So if we set our curvature to 0, then we get a constant growth throughout gyri and sulci. If we set it negative, we get increased growth in sulci and decreased growth in gyri. And if we set it to a positive number, we get increased growth in gyri and decreased growth in sulci. So we can explore these different cases. So here's some images of simulations that we included in that paper. So what I think you can see is that the overall morphology of these are pretty similar, but the thickness varies, and you can see that the growth varies too. So here in the preferential sulcal growth, you can see these pockets of high growth buried in the sulci. And here you can see, that the visible gyri have much higher growth. And then in this homogeneous case, you can see that the whole cortex is growing at the same rate. And so from this, we can calculate the normalized thickness in gyri and sulci separately. And so here the gyri are in red, sulci are in blue. You can see the 3 different cases that we considered. And we can also calculate that thickness ratio over time. And if we do that, so you can see that the uniform growth case, like we've seen before, it goes up and then sort of plateaus. In the case of gyral growth, that thickness ratio goes up and sort of keeps going up. And then in sulcal growth, it goes up and then comes back down. And that just so happens to like align pretty well with the data that we reported from the human brains in that PRL paper. And so I was like, awesome, my hypothesis was correct. I'm so great. Like I saw this coming. I just guessed really, really well. But actually we sat with this data for a little while and started to sort of question it. So we had to go back and reevaluate our hypothesis. And what we realized was we were comparing apples to oranges here. So we were comparing discrete and continuous measurements. In our simulations, as we had done in the PRL paper, we were comparing, we took eyeballed, the gyral peak and calculated the thickness of that, and the sulcal fundus, and took a ratio of those two measurements. So the peak and the max and the min, basically. And this is what we've done with our simulations as well. However, in the human brain, we were using this, basically dividing the entire surface into either a gyral region or a sulcal region, calculating the mean thickness of the gyral regions and the mean thickness of the sulcal regions and comparing those two. And so the difference was sort of, for this discrete comparison, we were calculating this thickness versus this thickness, which we ended up introducing this concept of coverage, which is a way to ensure that we're being consistent in our sampling. And so that was a very low coverage, like 7%, versus in our human data analysis, we had a coverage of 100%. Every point in the simulation or in the surface was either gyrus or sulcus. And so that was giving us a big difference, basically. So Schwillen introduced this consistent way to, sample the data. And we did this consistently between our simulations and between our human data. And we found that it made a pretty big difference. So basically, as your coverage increases, this thickness ratio is going to decrease, because instead of comparing the max and the min, you're comparing, the slightly larger numbers with the slightly smaller numbers. So we'd previously been comparing, this 100% coverage out here for the human data, which is in green, with the 0% coverage data from our simulations. And so that was causing this perceived agreement between these blue sulcal growth simulations and the green data. But really, when we are consistent out here, especially out here at 100% coverage, we see much more agreement between the gyral growth simulations and the human data. And so we can actually just sort of cut this data out here and look at it this way. And so you can see, again, this human data with the darker colors representing a narrower confidence interval. And then the red dots are showing our gyral growth simulation. So we varied a little bit, the stiffness ratio and the point at which we evaluated the thickness ratio. So that's what the variation gives us. So anyways, after we introduced this consistent sampling method, we found that actually the increased gyral growth best matched the human data. So it was a big lesson, I think, for us in consistency, which is, of course, a natural thing to, an obvious thing to include, but it's sometimes not obvious how to apply it to a given set of data. So in conclusion, if you could remember anything from this talk, remember that cortical thickness varies across the brain, and that thickness differences are a natural consequence of folding in soft-saw systems, but that increased growth in gyri likely amplifies this mechanics-based thickness difference. So some of the questions I was told to sort of expect, and maybe you have more questions about this, but I'll just briefly mention some future directions. We're interested in also looking at tangential folds. So gyri and sulci are not, we think, a strict binary, but rather a spectrum. We also have gyri and sulci that sort of bend and fold along their path. And presumably those folds also contribute to thickness differences. So we have a paper in submission right now looking at this sort of, and we do see that regions that are concave basically in both directions are the thinnest, and regions that are convex in both directions are the thickest. And saddle regions, which are part convex and part concave, are in the middle. So that's kind of interesting, supporting our ideas about tangential folds. We'd also really love to be able to remove this effect of mechanics. This is another thing that Bach had sort of hinted at. So he said in the future, we will need to investigate which of these areas are simply curved cortical sections to which no physiological meaning can be assigned. And he had some sort of words of warning. This critical correction will be an enormous undertaking, which, if it is to be completed with sufficient exactness, will be extremely demanding of the methods employed to achieve it, but we're going to try. And then we're also interested in how these patterns change throughout development, as the brain is folding, throughout phylogeny, or evolution in different brains of different species, and throughout health and disease. What do they look like in different disease or disorder cases? So just to acknowledge, again, most of this work was done by my postdoc Shuilin Wang and my graduate student Nagyahan Demirci, and our funding from NSF. And if you're interested in the papers that I talked about mostly today, you can find those there. And I'm happy to discuss further now, or if you want to reach out by email or on Twitter. So thank you very much.
Thank you, Maria.
Hi, Adrian.
Hey, I made it.
Great.
Actually, pretty much from the beginning of the talk. I was just maybe 1 or 2 minutes late.
Great.
All right. Yeah, that was great. I definitely made it for the quiz. And I got it right.
Did you get it right, though?
I got it right because I had seen that one before. So
yeah, fair.
I did study. Questions for Maria. I'll open up for the audience for questions. And I'll try to see if I can see people raising their hands. How do I see that? Oh, I see now the list of people. Yeah, so questions for Maria. Yeah, I see someone raise their hand. Mariana, do you want to go ahead and ask?
Hi, I was running on the treadmill while I was listening to your talk. Sorry.
Good for you.
It's all about double duty. The curvature that you're using for the model, what do you think it's a surrogate measure of a mechanical factor? You know, like what are the loading conditions on the folds that might, I'm just kind of wondering, like, to me, it makes sense that, I really appreciate you saying you're not saying the cells see the curvature.
Yeah.
But does the curvature induce a mechanical state that the cells might see that might be really related? Does that make sense?
Yeah. So if I go back to like my original ideas, oh, I think I just stopped sharing, didn't I? I can't go. Okay, let's see. Let me just share the keynote. So if I go back to the ideas that I was originally thinking about, where was that? Here. So can I zoom it out? Okay. So yeah, like I said, we do see regions of, in like a given, gyrus or sulcus, we do see regions of, this would be compression and this would be tension. And so I think maybe if you think about a gyrus, like maybe the inner property, like what the inner surface is feeling, is like more important to the behavior of the cells, because they're gonna sense like what they see first. And so maybe sort of mean curvature we're just using as a proxy for like the stress state of the inner surface. That's one idea I have, but that actually supported, my interpretation of that at least supported my sulcal growth hypothesis, not what we ended up finding with gyri, with the gyri. I think, so like I mentioned, there is evidence of differential gene expression in locations that will in the future be gyri and sulci. And so it could be that we're sort of using a kind of mechanics or geometry based surrogate for what is really like gene expression, and maybe we should try something that has like a different field, that we have a field of like, gene activity, and then growth is dependent on that. One thing that I think is neat about this model is that it's, as far as I'm aware, the first sort of responsive heterogeneous growth in the cortex, by which I mean we didn't predefine like this region is going to grow more and this region is going to grow less, which is going to affect the patterns that result. Instead, it's responsive. So it's homogeneous growth until a gyrus begins to form, and then it will grow like a gyrus. So that's why I think the curvature was kind of neat, in a way that I wasn't as excited about like a pre-patterned like field of gene expression, but that could be a future direction. So I don't know if that answers your question.
No, it does. Thank you. Great work.
Thanks.
All right, we thank you so much for the question. We have a couple more questions. Emma, I think, raised her hand first, then Kyoko, and then Johannes. So I can go ahead with Emma's question.
Yeah, thanks, Adrian. And thanks so much, Maria. That was a really nice talk. So I just have sort of like a maybe like future directions question for someone who's clearly thought a ton about this problem. Have you thought about how this might be relevant or sort of how kind of like secondary instabilities or different forms of instability, such as creases, might kind of like be related to this sort of framework that you've been thinking about with comparing the difference in thickness in different regions?
So you mean like if we see sort of, where do I have? I don't really have that slide in here. I was just working on another presentation where I had it. So for creases, and really in the brain, we see sort of like this folding, whereas in a lot of, at least very simple analytical models and some computational models, we see sort of sinusoidal folding. And so is that what you mean? Like the difference? Well, I guess we really have it in sort of here. So this is more sort of creases and this is more sinusoidal folding. And so if like the difference between those affects the thickness ratio, is that your question?
I guess it's sort of like, so for example, on this slide right here, if I'm interpreting it correctly, it looks like there's period doubling going on, at least in like the left column.
Yep.
I guess I was wondering if you thought a little bit about how like the emergence of period doubling might interact with the curvature dependent growth and how that might sort of like play into your model.
Yeah, that's a really great question. And I believe in this PRL paper, we have a slide that, or an image that shows sort of this variation in thickness ratio. And we actually see sort of two big increases in gyral thickness, and they correspond to the points of the initial instability and then the secondary instability. So I think that sort of second period doubling or secondary instability is sort of an instability superimposed on another instability, right? And so it has this sort of magnification effect in general.
Okay. Yeah, thank you. Yeah, I appreciate it.
All right. Kyoko, yeah.
Hi, Maria. Very nice talk. I'm home with the baby right now, so baby's joining. I was also, like Mariana, I was riding my bike at the beginning of the talk and I joined afterwards.
Wow, triple
so
tasking.
multitasking to the max. So I had a bit kind of just basic question about like what the brain material is like. So how porous is the material? Because I was thinking about folding, and if it's like porous, could it be that like the pore sizes are changing, and would that change the mechanics, like fluid pressure kind of thing at all?
A really good question. And there is a large body of work on like poroelastic models of the brain, especially in impact and injury. So like in the TBI community. And so I know that there's a large body of work. I'm not super familiar with it. So something like the size of the pores, I'm not sure about. Generally, we haven't included sort of poroelastic behavior just because of the time scales, like for fluid motion, like that's happening on a very different scale than growth is. But your question about pore sizes is sort of interesting because I think that was sort of, like I said, the intuitive. I thought it was intuitive explanation for the sulcal growth that like the analogy I made, if you're in a crowded room, you're not going to go into the heart of the crowd. Like any intelligent person is going to go sort of find a way through where there's some space, right? And so it made sense for me that might happen. And I would expect that to happen at sulci where at the bottom where there's tension. So it's very interesting though. Sort of the general question, there's also, I guess, evidence that sometimes cells will migrate towards stiffer substrates, which compression can induce stiffness increases. So that's sort of counterintuitive to me, but there's evidence that happens. But the concept of pore size, which I think of being as related to how much space is there for me to move, is interesting. So maybe some findings from or methods from pore elasticity could help us with that.
Yeah, it's a very Columbia question of me, I guess.
Thank you.
And then I guess I had one more question. So I was thinking about, you're looking at the developing brain, and I know that like there's a lot of like cerebral palsy cases, and apparently they're linked to like strokes that babies might have while in utero. And so is there a way to kind of like think about that problem, like blood flow? And I don't know if like blood flow is different from like general fluid flow in the brain. I don't know much about like brain tissue in general, but do you think that there could be like a mechanics based thing? Because I was thinking about, you know, where is it easier to flow into, right? Like where can cells migrate into? Is that something that
sort of being related to where blood could flow?
Yeah.
Okay. I guess the other thing that your question made me think about is like if blood flow is stopped to one part of the brain, that's presumably going to affect the further development in that part.
Yeah.
So yeah, I don't know anything about that, but it's an interesting question. So I appreciate that.
Cool. Thanks.
Thanks.
I was actually going to ask basically that exact question about the distribution of the vasculature throughout. And just to kind of add a comment, it would be interesting if there is a way to maybe look at cerebral microvasculature and if there's a preferential direction almost, if there's more vasculature, let's say in the gyrus than in the sulcus, and maybe that can direct some of the cellular processes one way or another. But
yeah,
I'm just curious.
I believe in this Bach 1929 paper, so there's also a section where he looks at neuronal arrangement and like sort of size and shape as an indicator of sort of what's the tissue doing. And I think he also looked at vasculature, and so the arrangement of the vasculature in gyri and sulci is characteristically different, like systematically different. So maybe that's sort of related. But yeah, I don't know anything about it beyond what I think his mentioned in that paper. So I wrote it. I have a little post-it note here that I'm writing down ideas to think about. So I appreciate it. Thanks.
Yeah, I mean, I wonder if you could embed sort of a very simple vascular network type simulation, basically, inside your folding simulation. So if you start out with a randomly oriented network, how does that sort of naturally evolve as these folds start to develop? I don't know if that would be...
Yeah, that's a good question. Thanks. Yeah.
Karen, I think you have your hand raised.
Yeah, thanks,
Hi.
Adrian. Hi, Maria. How are you? Good to see you. I love listening to your talk. That was great. I'm going to ask a very me question. So I'm wondering like how different constitutive models might affect your results. I know we don't have very good, constitutive models for brain tissue, but I'm just wondering like if you used a Neo-Hookean versus like some other constitutive model, like for like a general, modeling to a general value of stiffness, how that could change how it folds, if that makes sense.
Yeah, that's a really good point. There's been some evidence so like I said, there's still a lot of uncertainty around the properties, but people are working on it, very smart people. And I've seen some evidence that the Neo-Hookean model, which is what we use here, is not the best for brain tissue. And again, here we have the challenge of like different, so some of this work is being done like in the TBI community, and so maybe those material models aren't appropriate, but still even in like quasi-static sort of testing, probably Neo-Hookean isn't the best. And I think there is like a compression tension asymmetry in the Neo-Hookean model, which I think early on was at least part of our hypothesis for sort of, okay, but how is mechanics developing these thickness differences? So it would be interesting, and we haven't done anything with any model other than the Neo-Hookean, but it would be interesting to do. I mean, it would be very simple to run like the same simulation with just a different material model and see how much difference that makes. So yeah, I appreciate it.
Think, yeah. I do think your tension compression asymmetry point is like spot on, because you have both, right? And
Right,
so
and
I don't,
that's like exactly the sort of interesting part of buckling and bending is that you have both. So
Great.
there's cases where it's maybe not that relevant, but we definitely have both. So yeah.
do have people try to even make models of material behavior accounting for tension, compression, asymmetry in the brain?
I'm not aware of that. So I've just seen like work, considering a subset of existing material models like HGO and Mooney-Rivlin, and seeing sort of which of those best fits the data. But if you're talking about like sort of developing a model that would be like specific to the brain or specific. So I also hear, I've heard comments about the tension, compression, asymmetry, and Neo-Hookean, but I actually don't know if that problem exists in like all these other models that have been considered. So I haven't thought about it.
Okay, cool. Great talk.
Thanks.
All right, Johannes, you lower your hand.
I know I always want to give others the opportunity to share before I talk. I'm bouncing. First off, doing this work myself, I think it's always a lot of work that goes into just analyzing any model that you have in a way, or deriving the tools to be able to analyze is a lot of work. And I definitely like how your group is really good at visualizing the results. I'm bouncing back and forth between two questions, and so I'll try to actually ask both coherently, and then maybe you can choose which one you go after. The first one is the slide number 12, slide number 12, where you show these two the experimental results. If I remember correctly, yeah, this one, yeah. So I think if I remember correctly, the way they're done, they are produced, these images or these results, is very differently than how the brain evolves. So I think they sort of the substrate is pre-stretched, then the top layer is adhered, and then they are released.
Exactly.
You have, I think that's how they were prepared, right? Those samples. And so that is a very different mechanism than sort of a continuous evolution of a folding brain. And so I'm curious if you think that the conclusions you draw from this, how accurate are they, or how well do they match the outcome of a brain? That was the question number one. Then the second question is, and I'll try to keep it short, not to forget the first one. What is the, does this asymmetry in the thickness, or not asymmetry, but this difference in the thickness, when does that show up, do you think, in the folding brain? Is that something that emerges from the beginning, or could that be something that happens once the major folding is done and you have the proliferation of dendrites? So a much less severe mechanism, as in like neuronal migration. So saying that dendrites, so much smaller units than neurons, actually sense this compression or stretch and then just populate more.
Yeah, so two really great questions. The comments about pre-stretch versus growth, absolutely. So like I published a whole paper in 2017 in JMPS exploring the differences, like the effects on the instability if you have pre-stretch versus growth versus like whole domain compression. And they're similar, and in the literature they've been treated often as like the same, but they're actually subtle differences. And so very much agree that they're different. So I think, like I said, well, we had seen this lack of sort of quantitative agreement in this paper. And so what I would say is like the main conclusion I drew from this paper, the assertion that I would make is that mechanics alone, in the absence of the mechanics of folding or buckling alone, in the absence of any source of biological heterogeneity, can lead to thick gyri and sulci. And we show this using simulations and the experiments, and also a sort of theoretical analog. And so I am confident in that assertion. And I would actually say that using pre-stretch and growth and theory sort of makes that assertion stronger, because it's like a natural consequence of this like buckling of these soft materials. But absolutely see your point that like if we only had the experimental evidence and we're sort of especially making like quantitative conclusions from that, that I think it would be more troubling, like less sturdy to stand on. But I think that this does, add another sort of plank to our platform that mechanics is doing something in these instabilities that's or I guess, yeah, something non-biological is doing something in these instabilities that's leading to these thickness differences. And yeah, since our simulations pretty much only have mechanics, like I'm not sure what else that could be. Well, in our experiments. The other question, when do these thickness differences emerge? So yeah, it's a good question, and I'd really love to get some like good fetal MRI data and look at it. And I know you've done some really cool work like mapping different stages of those fetal brains onto each other, so you could really compare like, okay, we found at week 36 there's a gyrus here. What did that location look like at week 25? Was it already a bit thicker? So that would be really, really amazing data to get. And the registration is an important question, but also like the resolution of the scans, so those are big challenges for us. But also I will say in this paper, we had a very helpful reviewer who, pointed out that so my explanations had been sort of, the neurons migrating outward and they're looking for a less crowded space. And that again supported my incorrect hypothesis and not what we ended up concluding. And the reviewer pointed out that really folding happens after neuronal migration is complete. So it's so we talk about preferential sulcal growth or gyral growth and not cell migration preferentially towards sulci or gyri, because it likely is proliferation of those dendrites and different sort of remodeling that the neurons undergo once they're out in the cortical plate, and not the migration. So so I would expect, given what we now believe to be the case, that there's both biology and mechanics, that mechanics would contribute some cortical thickness differences, and then after the folds have formed, as this dendrite arborization is taking place, that the thickness difference would continue to increase. But yeah, we don't have good fetal data right now. We do have a 2020 paper that looked at children scanned around birth and then longitudinally at 1 and 2 years that shows that the cortical thickness difference continues to increase during this time. So that could be in support of that.
Thank you very much, Maria.
Thank you.
I have one question that we kind of sent on the list, maybe to wrap up. What was the most challenging part of this study, and I guess particularly also for students in the call? Yeah, where were you stuck the most when you actually were doing the paper?
That's a good question. So for me personally, like I was sort of stuck on this, and I didn't know how to do this. But my postdoc Shvalun, who I've mentioned a couple of times, is just a really, really excellent computationalist and is sort of a wizard in abacus. And so he, created this growth model. So I mean, I put one equation and like sort of one built up slide in here, but obviously that was a huge amount of work to come up with the equations and come up with realistic values for the parameters and write the user subroutine for that. So I think that was a pretty challenging step, just sort of getting over that initial hurdle of being able to do the simulations. I don't know, I think Shvalun is on the call. I don't know if he has a different addition.
Yeah,
maybe it wasn't that hard for him actually.
Yeah, I think the numerical implementation is relatively easy for me. I think the biggest obstacle is to find the consistent comparison between the experiment and our simulation. That one is it's a little bit hard for me. Yeah, I think.
Yeah, so in general, especially I think Shvalun, as he mentioned, the numerical implementation was fairly easy for him. So, you know, I feel like his ability to simulate things has often outpaced our ability to find reasonable and meaningful basis for comparison to like experimental or imaging data or something like that. So in my experience, he can sort of simulate anything that I can dream up, but we have to sort of, we don't want to fly too close to the sun and you know, go beyond what we have data to sort of support and validate and things like that. So
Awesome. That was very, very insightful. Thanks for answering that.
yeah.
Yeah. Any last question from the audience? Otherwise, I think we are just coming up on the hour. So I guess I can officially conclude this very, very good session of Closer Look. Thanks, Maria, again for the talk.
Yeah, thank you all for having me, and thanks for coming. It was really nice to talk about this, and great questions.